Hi, my name's John Webb from Natural England, and over the next 3 days volunteers and Natural England staff will be collecting insects and passing these over to the Natural History Museum, who will be extracting genetic information from them. This is cutting edge science which will make biological surveys in the future much more easier.
Hi, I'm Mike Downey and I work with Natural England on national nature reserves, and it's fantastic to partner up with the Natural History Museum for this DNA Bioblitz because we share a purpose for conservation of the natural world, for science and for engaging the public in our natural world. And here we are at Ainsdale Sand Dunes National Nature Reserve. It's a fantastic site. It's a hotspot for biodiversity with a great range of habitats for this sort of new pilot approach to DNA sampling. So great to partner with the Natural History Museum.
I'm Gavin Broad. I'm in charge of the insect collections at Natural History Museum, and I'm here with several of my colleagues, and we're trying to kickstart our involvement in the Darwin Tree of Life project. So this is a big collaborative project where we're going to try and sequence the genomes of every UK animal and plant. So it's quite ambitious. This is an amazing sort of opportunity to collaborate with Natural England today. So we've got experts sort of coming in from around the country, and we're going out there collecting a lot of insects, a lot of other invertebrates, and we're going to sequence them in real time. So we're going to go from field collection to a DNA sequence by Thursday. We're going to add a lot to our knowledge of UK insects and other invertebrates, and we're going to forge some good links. One of the things we're going to get out of this is genomes, hopefully. So we've got specimens that are being flash frozen. We're also hoping to build a big DNA reference library. So this is DNA barcode sequences, cytochrome oxidase 1, and we're generating those live now. We've got experts on moths, we've got amphipod crustaceans, wasps, that's me. We've got some people who work on spiders, loads of dipterists looking at flies. We've got an amazing bunch of people out here today.
Hi, I'm Andy Briscoe from the Natural History Museum, and I'm here at Ainsdale as part of the DNA Bioblitz, where we're attempting to sequence DNA in the form of molecular barcodes from all of the identified specimens that we collect here, so that we can generate sequence data that can be used to rapidly identify these things in the future for parts of other sequencing efforts like the Tree of Life project or other bioblitzes and inform biodiversity assessments.
Hi, I'm Andy Hall. I'm from the Natural History Museum, and I'm working here at Ainsdale in the pop-up lab, and I'm here to tell you the technical bit. So the specimens come in, and I take a tissue sample from each one and place them in a well of a 96 well plate. And I then add an enzyme called proteinase K, and the proteinase K digests the tissues to release the DNA. I then add a resin called Chelex, which binds to all the junk that we don't want, and that then sinks to the bottom of the sample, leaving the DNA floating in the liquid at the top. We then take the liquid from the top and we put it into something called a polymerase chain reaction, and we use this reaction to tag the DNA in a specific place. We're looking for a gene called cytochrome oxidase 1, which is known as the barcoding region, and we tag that region with synthetic bits of DNA which have indexes on the end. Once we've done the polymerase chain reaction, we then pool all of them together into one tube, and we then run those on the Oxford Nanopore MinION, and that sequences the DNA. It sequences all of the strands, so we get an awful lot of data out, and because we've tagged each one with an index, we can then tie that back to the original sequence, because each sample is tagged with a different index, and that allows us to get a sequence for every single sample that we process, but just in one sequencing run.
So hi there, over the past 3 days we've collected genetic information from Ainsdale and the River Ribble, and that's included 55 citizen scientists, agency staff and museum staff. I'm going to have to look at my clipboard here to tell you exactly who's involved. That's included the River Ribble Trust, Lancashire Wildlife Trust, RSPB, Buglife, Edge Hill University, Liverpool Hope University, the National Museum of Scotland, the North and East Yorkshire Ecological Data Centre, Environment Agency. So thank you very much to all those folks, and it's been led by the Natural England and the Natural History Museum, and it's included specialists who've looked at moths, bugs, beetles, that's me, wasps, that's him, and many more groups. And over to you, Gavin, for some results.
Yeah, so all these people together have been sort of bringing in samples regularly, and we're up to about just under 1,000 specimens database so far. We've had the first 384 legs have been sequenced, so they're going to produce about between 1 and 1.5 billion base pairs of genetic information, so it's an incredible result from basically 2.5 days work.
It's been absolutely fantastic hosting the world's first DNA Bioblitz here at Ainsdale Sand Dunes, using national nature reserves for what they were designed for as outdoor laboratories, and it's been really great to see the amount of effort that has gone into this week. We've had 14 different partners, including our friends at the Natural History Museum, and lots of local organisations involved as well. We've had 59 different participants getting involved in the survey effort and in the laboratory work. We reckon we've travelled around about 170 miles on foot over the last 2.5 days, and our highest Twitter count was 4,160. So let's see if this one beats it. Thank you all for getting involved.